Now that we have a basic foundation for evolutionary theory, we can look at evolution as microevolution and macroevolution. Today, now we're going to be looking at microevolution, which basically is a change in gene frequency. Now, if you remember, the gene pool or gene frequency determines, of course, the type of the characteristics that a given organism is going to have. Within a population, if that gene frequency changes significantly, you're going to slowly shift over and become a little bit different, and eventually may become a completely different species. Let's look at some key terms. Species, we all know that species are organisms that can mate. Populations are basically a group of organisms that live in a given environment. The genotype and phenotype, the genotype, of course, is the genetic components of a given organism. The phenotype is the expression of the genetics. Mutations take place, of course, within the DNA itself. Alleles we refer to as DNA pieces that make up genes. Now, this is the important point I want to make here. Gene pool. The gene pool consists of all of the alleles in a given population. If we're all in a classroom together, and if I could take all of our genotypes and categorize them carefully, I would say this is our gene pool for our class. Our gene pool reflects the characteristics that we have in this particular population in this room, let's say. Now, if the gene frequency changes significantly, the population would shift. If the gene frequency changes significantly, then the characteristics of this group would change also. Let's think of this example. If all of us in this particular gene pool were to leave and go to a far-off planet and reproduce, our gene pool would change, and 10,000 years from now, our gene pool will have changed so drastically that we probably would evolve into a new species. Okay, so now we understand what a gene pool is and how gene frequency change results in basically microevolution. So, to summarize, microevolution are small changes in the gene frequency in a population. Let's look at microevolution within a given species of frogs. If we have a population of frogs that exist perfectly happy in an environment, they're able to, of course, to mate, to reproduce, and we maintain a certain gene pool. If a barrier develops that suddenly keeps a subset, population A, from mating with population B over a period of time, population A will evolve to look different from population B. Why is this? Because the environment in population A poses different challenges. The environment in population B poses different challenges. And as a result, population A will evolve into a species that can meet those challenges, and population B will evolve into a species that can meet the challenges in the area that we're looking at. So, as a result, we have potentially two distinct populations resulting. So what is the genetic basis for microevolution? Which is basically, we are saying, changes in the gene frequency. If we look at the characteristics that evolved in the previous example, we see that the characteristics were color. A lighter color met the challenges in one area better than a darker color in the other area. Color is determined by different genes, a pool of genes. As we can see, dark coloration was due to gene A and A2, and light coloration was due to gene A2 and gene A4. So in the evolution of two species, we see how the alleles or genes essentially migrated, and whichever combination of genes allowed that organism to better meet environmental challenges, that population survived and did well. So what are the forces that change the gene frequency and drive microevolution? There are a number of things that we need to look at. Mutations, of course, take place at the DNA level. Gene flow, we'll talk about gene flow. Genetic drift, non-random mating, and natural selection. So let's look at each of these. Mutations, as we said, take place at the DNA level. Mutations may be point mutations, or mutations may be a whole piece of chromosome gets lopped off and we have a deletion. Of course, any change in the DNA, any change in the genotype will result in a new phenotype. Now, gene flow is the movement of genes from one population to another population. In this example, we see that the silver sword tarweed had migrated from the mainland and moved over to Hawaii. On the Hawaiian Islands, this new organism had new conditions to compete. So this new organism had a different environment to grow in. As a result, evolved into a new species. So the point of gene flow is that it results in a change in gene frequency. Now, genetic drift. Genetic drift is another way of changing gene frequency. Now, genetic drift usually results from disease or natural disasters which reduce the population to a small number. Now, in this slide, we're looking at a bunch of penguins. And if we look at the red penguins, they represent the gene frequency of red penguins. Now, to the left, you can see that if a natural disaster comes in and wipes out half of the penguins, the gene frequency doesn't change very much. It goes from 10% to 9% for the red penguins. However, if we deal with a small population of penguins to the right, and a disaster comes in, look what has happened to the little red penguin. It's gone. The gene frequency for red is gone because the population is very, very small. So small populations are always in danger of losing vital, important genetic material. Other examples of genetic drift is the bottleneck effect and the founder effect. Now, here we're looking at some sea lions that are fairly rich in their genetic pool. Now, when hunters come in and kill off most of these sea lions, you see that the genetic pool, which is illustrated by different colorations of animals, becomes very small. Now, once these animals move on and start to reproduce, you see that the end result is a population that is less rich in genetic diversity than the original population. This is referred to as the bottleneck effect. The founder effect is also an example of genetic drift. For example, if a group of individuals decide to move from one area to another area, they're carrying a small sample of their genetic material. So when this happens and the new colony is formed, they have less diversity and therefore they're much more subject to disease, as is exemplified by they're much more susceptible to disease. Now, if I ask all of you in our population, do we have random mating or do we have non-random mating? Some of you will probably give me a very surprising answer. Reality, folks, is that in nature you have non-random mating, which means that organisms within a given species are programmed to select mates that have characteristics that will pass on their genetic material and assure the success of the species. In nature, we have absolutely non-random mating. So, the bottom line to all of this is that natural selection is the last force that drives microevolution. Organisms respond to the environmental challenges by adaptation, and this is, of course, the major force that contributes to evolution. So this now leads us to some philosophical questions. We ask ourselves, is there a purpose to all of this? I'm not sure there really is a purpose to all of this. Evolution really has no blueprint. There's no plan that scientists can envision. Basically, evolution is not a movement towards perfection. Evolution is simply a process that responds to the environmental challenges and basically is at the mercy of genetics. Now, we do have three modes of natural selection: stabilizing, directional, and disruptive. So let's look at each of these. In stabilizing selection, a population can be looked at to your left as you start off with a broad curve and end up with something towards the middle. In directional selection, we can look at characteristics that tend to move either to the left or to the right. And in disruptive selection, a given set of characteristics will kind of break down in two so that you have one set of characteristics that somehow or another manages to survive, and another set of characteristics that manage to survive pretty much in the same environment, because both set of characteristics can compete effectively in the particular environment. So if we look at, again, a stabilizing selection, we see that infants tend to fall within a certain birth weight, because being born too early is no good and being born too late is no good either. And this is the way nature works. In directional selection, we're looking at a process that evolved a particular hard shell clam. As you can see, that this particular organism started out as being very smooth millions of years ago, and now started out as being very rough million years ago, but somehow the characteristics of being rough became smoother and smoother, and apparently for this particular organism, a smooth shell is more advantageous than a rough shell. So you see, here we have directional selection. And disruptive selection is shown by the top group of birds here have wide beaks, the bottom have narrow beaks, and they both seem to get along quite well in their particular environment. So this particular slide basically summarizes the different forces that drive microevolution, from mutations to gene flow, genetic drift, non-random mating, and natural selection. And I won't read the slide to you, but it's a very, very good summary for you.
And my LP comes with a foolproof warranty. If you buy it, don't like it, feel free not to listen. I'm not sure about a refund, but you're welcome to diss it, 'cause I